Bây giờ mời bạn Ray. Ray, please, your turn, please.
Yep. Let me share my screen.
Thì bạn bạn Ray này là đại diện cho Bắc Bio, và bạn sẽ nói về cái short list hôm nay. Thì mọi người chịu khó nghe tiếng Anh đúng không? Lâu lâu mình cũng học nghe tiếng Anh đúng không? Đó. Xong sau đó rồi thì mình sẽ dịch lại sau, hoặc là có câu hỏi sau thôi mọi người ha. Rồi, Ray, please share your screen, please.
Yep. Let me give me 1 second.
Ừ.
Let me. It asked me to read. Okay, okay, okay. I think I can share now. Okay. Yep. Give me 1 second. Let me bring the screen up. Okay. Uh, thank you so much, everyone. Good evening. So, thank you so much, Dr. Loy, for inviting me to provide a presentation on, talking about PacBio for how we actually advance microbial genomics with our highly accurate short read and long read sequencing. So, just to introduce myself, so my name is Ray. I'm the territory account manager for PacBio, taking care of Southeast Asia. So I will start by, giving an introduction about our new short read system, which actually set the new bar for all the for the new benchmark for the high accuracy for short read sequencing. So yeah, so we launched the ONSO system, in the late 2022, which significantly changed the benchmark for accuracy for short read sequencing. So if you do see here, we are able to actually achieve, more than 90% of our bases, which are above Q40 plus, which means that, they are 99.9%, 99.99% accurate, or for the reads that are actually generated from our sequencer itself. And what actually enabled this highly accurate reads is actually from our chemistry, which is called the sequencing by binding chemistry. So if you can see here, so this is our SBB chemistry, which how it actually starts by, the polymerase will actually initiate the sequencing process. And on the DNA strand, which is growing, we actually have a nucleotide, which has a blocker. So to prevent any additional bases from actually incorporating. So during the second step, right, we will actually flood the flow cells with fluorescent tag nucleotides. But you can see the blocker is still here. So when the fluorescent nucleotide is actually attached to the complementary strand, right, it's only attached temporarily. And then the optical system will actually, capture the signal of this particular fluorescent. And then after that, it will actually be washed away. And then after that, there is a process to actually, remove this particular blocker. And then we will actually incorporate a new nucleotide with a blocker, but without the fluorescent tag. So, with this particular chemistry, right, you can see the difference between, SVB and SBS is we actually separate this particular process out into two, which is this interrogate and incorporation process. And by doing that, right, we are able to actually achieve the benefit of getting high accuracy for our bases to achieve Q40. How do we do that, right? Is because, the fluorescent tag nucleotide is not directly integrated to the DNA strand. So this helps to eliminate any negative effect of molecular scarring, which will actually occur. And then this will actually increase the molecular scarring will actually cause the increase for signal to noise ratio, which eventually will affect the accuracy of the reads. So with our SBB chemistry, right, we are able to actually overcome these limitations that are present in other, NGS method by, and in the end, we are able to actually generate reads that are very accurate. And then we can also sequence through, difficult and repetitive regions as well. And then unlike other NGS system as well, we have minimal index hoppings as well as duplication rate in our ONSO system. And then by having high accuracy as well, we are able to actually lower the input requirements to achieve the similar sensitivity. And by doing that, we are able to actually save on your precious samples itself. And together with our ONSO sequencing system, right? So we actually supplies the ONSO cycle sequencing kit, available in 200 cycle or 300 cycles. And then we also provide the library prep kits. So we have library prep kits for high molecular weight DNA as well as for fragmented DNA as well. And if let's say, your lab already has a library prep kit that you all usually use, or like a workflow that you already optimized and validated, we do have a conversion kit that can actually help you to convert your library to our ONSO compatible library. And then you can run it on our ONSO sequencer. And then you can also analyze with your existing pipeline as well. So with our 12, more than 12 years of experience, commercial experience for PacBio, right, we are able to actually provide very comprehensive service and support, as well as commercial excellence to our customers for the short read sequencing as well. And for our short read sequencing, it's very easy to use. So, it's, to load the flow cells to start the run, you only need less than 10 minutes. So it's very easy to actually, run the instrument itself. And we also want to actually provide an end-to-end solution to our customers. So we actually partner with our PacBio compatible partners, such as, 10x, Twist, Agilent, QIAGEN, to provide the library prep reagents to be run with our sequencer. And then after that, after sequencing as well, we do partner with bioinformatics company like Google Health, Sciention as well, for all the different secondary and tertiary analysis that is required to actually analyze your data. And okay, so I think the theme of today's talk is on microbial genomics, right? So I actually pick out a case studies of how we use our ONSO to actually help on microbial research. So one of the case studies that we have is actually we collaborated with Zymo Research in the US, and we actually use shotgun metagenomics to actually test on wastewater samples. So what we want to actually achieve is to actually detect the dominant species, as well as if there's any functional groups that are beneficial or harmful to the treatment, as well as if there's any presence for pathogens, as well as antimicrobial resistance gene in the wastewater samples itself. So we compared the performance between Illumina SBS chemistry as well as PacBio ONSO SBB chemistry. So if you can see here, so you can see here that the ONSO SBB chemistry actually was able to actually detect more microbial diversity. So we detect like 121 genera compared to SBS that only detected 72, and this is due to our higher sensitivity on our ONSO system itself. And we are also able to detect more AMR classes as well. So you can see that if you can see from the bar chart here, right? So you can see that for SBB results, there are more colors. More colors means, you are actually able to detect more different classes of AMR genes in the particular sample compared to the SBS one. So you can see in sample 1, sample 2, sample 3, we do have more colors and different categories of AMR classes that we detect in the sample itself. And in summary, just to summarize, for our SBB chemistry, due to our higher accuracy, we are able to actually detect, more species, as well as antimicrobial resistance classes and functional bacteria in, difficult samples such as wastewater treatment samples. And, besides, due to the high accuracy of our SBB sequencing chemistry, it's also can be used not only for microbial genomics, but also for things like cancer genomics, as well as application that requires high sensitivity. Let's say if your target of interest is very low in concentration, our ONSO system will be very suitable for this type of applications. Okay, so I think I've talked about our ONSO short read system. So next, I also want to just touch a little bit on our PacBio long read sequencing as well. So as what Dr. Loy has shown just now, right? So we have short read and long read as well. And PacBio is the only company that actually have both long read and short read. So we have our short read, the SBB chemistry, and then on long read itself, we do have our HiFi sequencing on the Revio system. So for our HiFi, so the basis of our long read sequencing is actually based on this chemistry called HiFi sequencing. So what HiFi sequencing does is we will always start from a high molecular weight DNA, and then we will actually attach the adapter on the SMRTbell adapter to the high molecular weight DNA. And then after that, we will actually sequence it through a few times to actually generate a lot of sub-reads. And then we will combine all these sub-reads to actually produce one read, which we call HiFi reads. And eventually, these HiFi reads will have very high accuracy. We can actually achieve 99.9% accuracy, and we can actually generate reads up to 25 kb. So as you understand from like short read sequencing, most of the reads that are generated are only 100 to 150 base pairs, right? And then they will use bioinformatics pipelines to actually, assemble them together to form a contig. But for HiFi sequencing, the raw reads that come out is already 25 kb itself. So this will be able to, help you to, achieve a lot of things that short read cannot do. For example, like structural variations, repetitive regions, pseudogenes, all these different regions, HiFi reads will be much more competent in detecting this kind of variants. And in terms of microbial genomics as well, with the high accuracy and long reads, we can actually support multiple different applications, from microbial assembly, epigenetics, viral and targeted sequencing, as well as 16S or shotgun metagenomics. And compared to short read itself, for microbial whole genome sequencing, we can actually get more complete microbial genomes with ease and confidence because our reads are so long already. When we combine them together, it's much more easier compared to the short read itself, and we are able to actually achieve closed genomes as well as to sequence through the plasmids as well. And we do not need any short read polishing for our microbial whole genome sequencing. For full length 16S sequencing, as I think just now during the first presenter, they showed there are different, you can actually sequence, use different method to actually sequence for 16S, either V1, V2, V3, V4, right? But for PacBio long reads, we actually sequence the full length from V1 to V9. And by doing that, right, we are able to actually detect down to species and strain level. So if you do V3, V4 sequencing, right, usually you will probably get taxa or genus level kind of information. But for full length 16S, you are able to, we are able to go down to species and strain level. And there are studies that show that, with full length 16S as well, the correlation that you get the data that you get might be similar to what you can get from shotgun metagenome profiling as well. And in terms of for shotgun metagenome, so for HiFi sequencing, we can do both profiling as well as assembly itself. And for our shotgun metagenome profiling, we can actually provide much more richer functional information as well without any error correction or assembly required. And we can achieve these high quality results with as few as only 50K reads, which enable you to get low cost sequencing. And talking about cost itself, right? So I think people will always think long read is much more expensive. But I think with the recent new instruments as new chemistry that we get, we are actually able to lower down our costs, for example, for 16S, as competitive as what short read sequencing can actually achieve. Yeah, and lastly, on shotgun metagenome assembly, we can actually provide far more high quality max. You can see here in the photo, this is very small here. So these are all, like close max like, all these metagenome assembled genomes that, which you can actually generate complete circular single contig maps with our long read sequencing. Okay, and I think this is my final slide to let you all know, with our new instrument right now, we can actually, scale up for microbial research by a lot. So for example, compared to our older system, which is the Sequel 2E system, right? We can actually generate, 10 times more shotgun metagenome profiling throughput compared to the Sequel 2E. So for example, if let's say we are doing shotgun metagenome assembly, we can run, around 4,000 samples per year. For shotgun metagenome profiling, we can, generate up to 120,000 samples per year. So, and this is just on the scalability itself. So we can actually scale the microbial research by a lot, but we do still provide flexibility in terms of the Revio, because we have 4 different stages. You cannot see here, but there are actually 4 different stages in the Revio, where the 4 SMRT cells can be analyzed together. But you can actually run different runs, different types of experimental design on it. So you can, in one SMRT cell, you can run 116S samples. In second SMRT cell, you can run a shotgun metagenome, and a viral amplicon, as well as a microbial whole genome sequencing in the third and fourth SMRT cell. So it's very flexible that you can run multiple different experiments in one single Revio run. Yeah. So with that, I think I would like to end my presentation and pass it to the next speaker. Thank you so much.
Thank you, Ray. And stay tuned. Wait until at the end we have the Q&A section. Yeah.
Okay.
Maybe have a lot of questions waiting for you. Thank you a lot, Ray. Rồi thì cái câu hỏi với lại câu trả lời tôi cũng copy từ trong cái phần chatbox ra giống như vậy. Thì không biết là mọi người có hài lòng, em cứ đi hết đi Huy. Tại vì mình không có thời gian nhiều quá để đi lại á. Đó, ở đây nếu mà anh chị em nào mà cần câu hỏi câu trả lời ấy, thì tụi tôi sẽ gỡ cái file này. Tụi tôi đã copy qua hết trơn rồi, bởi vì mọi người đừng có sợ là mình bị thiếu. Right, we copy on the question and the answer to the file online. So because we don't have time, so we don't go through every question. So over here is the answer from you guys, and then we copy it and pass into the file. Do you think it's okay?
Yep. Thank you so much, Dr. Loy.
Okay, perfect. Rồi, nếu mà không có câu hỏi gì thêm thì ở đây mình có thông tin của bạn Thiện ở bên DKSH là nhà phân phối của Bắc Bio. Còn Ray thì là trực tiếp làm bên Bắc Bio, là bên hãng. Đó, đây thì mọi người chụp màn hình lại đi. Còn nếu không thì tôi sẽ gửi cái link ở trong cái phần bài giảng của lớp của mọi người. Tôi sẽ gửi cái link để cho mọi người có thể contact được. Rồi, như vậy một lần nữa cảm ơn các bạn đặt câu hỏi, cũng như cảm ơn Thiện bên DKSH và cảm ơn Ray. Again, thank you so much for your time, Ray, and we hope that we can have more for a long read next time. Yeah, for the webinar next time. Thank you.
Yep. Thank you.